All right, welcome to fall 2023 Colloquium. Here for fall 2023. We have a candidate to be a genetics PhD program trainer today. We've seen seminars from all across UW-Madison this semester already. I think this is our first one from the vet school, so I'm excited to welcome you from SVN today. And I'm also excited because this is one of my favorite topics, replication and repair. But let me tell you a little bit about today's speaker. So I learned that today's speaker, Kavi Mehta, is a native of Wisconsin and did not one but two bachelor's degrees here, one in biochemistry and one in French. Anybody here? So then he made a giant move down south all the way to Northern Illinois, to Northwestern University, to do a master's degree and spent a brief time in industry, and then went back to Northwestern to do a PhD, working on HPV and carcinogenesis. Then he did his postdoctoral work at Vanderbilt University, where he was working began working on DNA repair. And so he's continuing to work on this in his lab in comparative biosciences over in the vet school. And one thing I don't know, when did you arrive here?
I started August 1st.
Oh,
yeah.
Well, welcome. Let's give an extra special welcome then to today's speaker, Kavi Mehta.
All right. Hi, everybody. Today, I'm going to talk to you about mechanisms of replication-coupled DNA repair. And a lot of this, of course, is my postdoc work, but I have a few snippets of sort of new data at the end of the talk that I think is quite interesting. And so to begin, I'm really going to talk about a brief background about the replication stress response. And so to begin, the amount of human genetic code is vast and it needs to be replicated faithfully. And over a lifetime, our cells undergo 10,000 trillion cell divisions. Printed in 12-point font, our genetic material would stack higher than the Statue of Liberty. And each cell really importantly encounters tens of thousands of lesions per day. And why this is important is because this can be from endogenous sources that are sort of byproducts of metabolism, like reactive oxygen species. Also things like exogenous sources like industrial chemicals, chemotherapeutics, other sorts of genotoxins, and things like oncogenic viruses. And so the interesting thing is that these lesions are diverse and they're repaired and surveyed by an equally diverse array of mechanisms to maintain genetic fidelity. So on the right here, you have alphabet soup. And if you guys are interested in DNA repair, I'm sorry if I don't have your favorite repair process on here, but basically why I put this up is that this is to show that there are this multitude, myriad ways of fixing lesions. And they include things like nucleotide excision repair, base excision repair, mismatch repair. But one thing that's really important to note sort of in this talk today is that many of them are regulated by these three master kinases: ATR, which responds to single-stranded DNA during replication, ATM, and DNA PK, which are more important probably for double-strand breaks. And so what's important, and the reason why we all study this, is that defects in DNA replication and repair and the stress response result in diseases, cancer, and aging. And so just an example of that, we know that oncogenes put cells under chronic genotoxic stress. This is by virtue of pushing them through the cell cycle when they don't want to be. And this chronic stress can induce genomic instability and mutagenesis. And importantly, defects in repair potentiate both oncogenesis and developmental disorders. So I put up three different genes that can have mutations in them or be deleted that result in problems. And I put a bracket here just to point out that BRCA is a really important DNA repair gene. We all know about it, but when it's lost or there's defects, it results in a higher propensity of getting breast cancer. And when there's a defect in ATM, this master kinase, you can get this disease called ataxia-telangiectasia, which can result in cancers and neuropathologies. And defects in ATR result in growth defects and microcephaly. So although the activation of the stress response is sort of well characterized, pathway choice and repair intolerance processes during replication are not. So in other words, what dictates which pathway is being used, particularly in DNA damage tolerance pathways, is not well understood in human cells. And so today's talk is really broken up into 3 vignettes. I'm going to talk about 2 papers that I published during my postdoc. The first in which I showed that this checkpoint regulator, CHEK1, which is a downstream effector of ATR, actually directly phosphorylates this very peculiar and specialized polymerase known as PrimPol for its actual reprimase activity. Next, I'm going to switch gears to this very peculiar protein known as hemces that was discovered in my lab, my postdoc lab. And it's peculiar in that it actually forms a DNA protein crosslink to protect DNA from replication stress. So it's actually forming a lesion in the genetic DNA to prevent double-strand break formation. And finally, I'm going to switch gears to talk about things that I'm going to be studying in my new lab here at the University of Wisconsin. So before I get started, I just want to cover the replication fork. And this is a very, very, of course, pared down human replication fork. Studies in many labs have suggested that there's upwards of 600 proteins. Actually, one of Jim Keck's, who's in the audience, PhD students found this in Dave Cortez's lab, that the human replosome is likely made up of over 600 proteins. Here we have the leading strand on the bottom. This process is considered a continuous process, with pol epsilon being tethered directly to the CMG helicase, which unwinds the DNA. The lagging strand synthesis occurs discontinuously through a series of repriming events through pol alpha primase, which first lays down a small RNA primer, and then a small DNA primer is extended from that, and then it hands off to pol delta. And here I have RPA just showing that RPA can bind single-stranded DNA and code it when it exists. So, as I said before, there are many options to deal with replication stress. And so I'm going to talk about just 3, and I'm really sorry if I've left out one of your most favorite pathways. But I just want to illustrate how there are different sort of scenarios and why these different scenarios matter. And so on the left here, we have a replication fork again, but in this case now, I put this star-shaped damage here. This could be a bulky adduct, it could be an abasic site. And so what happens, or what could potentially happen in human cells, although we're not that sure, is that the polymerase can reach this lesion, the CMG helicase can bypass it, and this leads to uncoupling of the polymerase epsilon from the CMG helicase. This leaves now a substantial amount of single-stranded DNA that's rapidly bound by RPA, the single-stranded binding protein, and this leads to the activation of ATR, as well as recruitment of ATRIP, one of its partners. And this can result in 3 possibilities. Again, non-exhaustive. With translation synthesis, there's this host of specialized polymerases that are thought to respond to specific types of lesions in DNA. And what they can do is they can just put a base right across from this lesion, or they can just bypass it entirely, extend, continue replication, which is the imperative of the cell. But this comes at a cost, and the cost is likely mutagenesis. And I say likely because we do believe that they're error prone, although the evidence in human cells that they in fact are is not wholly there yet. With pol repriming, this is the specialized polymerase that was discovered about 10 years ago. And this is really interesting in that it actually, I think, has adopted parts of the HSV primase, so human or herpes simplex virus, and it can actually reprime downstream de novo. So if there's a lesion, this polymerase reprimes on DNA downstream and then hands back off to either replicative polymerases or TLS polymerases. This again comes at a cost. The cost is that by this repriming action, it leaves a strip of single-stranded DNA that needs to be filled in post-replicatively. And this is thought to be done by translesion synthesis polymerases. So this process is also thought to be more error-prone than normal replication. And finally, I put up this structure here called fork reversal. And fork reversal is this really interesting method in which these series of motor proteins in human cells, which I'm going to talk about later, this is also seen in both bacteria and yeast, but effectively push or reverse the replication fork to put the lesion back in the context of double-stranded DNA, also causing annealing of the nascent strands. And what this is thought to do is this is thought to sort of buy time so that more error-free repair choices can be used, such as things like template switching. And it stabilizes this, of course, and puts it back in the context of double-stranded DNA. These are dynamic structures that can be reversed, and they can restart and reverse and restart. So today, I'm going to talk to you how this really particular protein in polymerase prim-pol is regulated by CHEK1, this downstream effector of ATR. But before I get started, I want to again hit home why this is important. And why it's important is because which pathway is utilized is important. So if you have normal replication go just fine, you get a nice copy, you're good. However, if you have prim-pol repriming activity, now you have a single-stranded DNA gap that's prone to cleavage. Translesion synthesis polymerases can induce mutagenesis, as shown here. And although I had said that this chickenfoot structure is, even though it's labile, it is a method to put the DNA back into the context of double-stranded DNA, it can still be cleaved by things like structure-specific endonucleases like MUS81 or SLX4, or exonucleases can degrade this nascent strand, resulting in genetic instability and the loss of genetic material. This is all under the context of what determines which pathway you use is not well defined. So in other words, not all replication stress is the same. It can be on one strand, it could be on both. It can be caused by endogenous sources, chemicals, exogenous sources, and even viruses. And this is really important to take into account because there's a substantial amount of information known about this in bacteria and yeast and also in vitro systems. However, very little is known about what happens in human cells. In other words, context is important. So to begin, I'm going to talk about the model systems that I'm using to study these events in my lab and what I use to study and answer questions that my ex-postdoc advisor was interested in at the time. And these were sort of developing tool sets to characterize the effects of lagging and leading strand replication stress in human cells. And so what I started out with is I fused a protein called ApoBec3A, which I'm going to talk about in a story a little later, which is a cytidine deaminase. It's also a viral restriction factor, and it converts cytidines to uracils. The uracils can then be excised by UNG, and it results in an abasic site. And importantly, these abasic sites that it forms are thought to be more prevalent on the lagging strand of DNA, suggesting, so this was a really good way to potentially induce preferential lagging strand lesions, where this persistent single-stranded DNA is. And I fuse it to this modified estrogen receptor so that it would be, when expressed, it would be expressed in the cytoplasm. It could be very rapidly shuttled into the nucleus. I then also adopted a recent discovered inhibitor called CD437, which is a direct catalytic inhibitor of pol alpha, this lagging strand polymerase primase. And I've recently developed two cell lines which allow me to very rapidly degrade both polymerase delta and polymerase epsilon, the leading and lagging strand polymerases, which I'm going to talk about later. And these are really, really sort of exciting, and I've produced some really interesting data with them. And so I'm just going to focus right now on this polymerase alpha inhibition and ApoBec to answer questions that I was interested in and what my postdoc advisor was interested in. And so to begin, I'm going to look at polymerase alpha inhibition. And really what I wanted to do was characterize what happens when you inhibit a major lagging strand polymerase post-initiation. And what I found was that short-term pol alpha inhibition really preferentially generates lagging strand single-stranded DNA really rapidly. And so I did this using a native BRDU assay. And what I found is that this amount of single-stranded DNA RPA signaling was through the roof within a very short time period. So I was really sort of interested in what would happen to replication in the absence of a major polymerase. And although this seems like a sort of peculiar question, it's been shown that even though this is a very, hitting the system with a hammer, that, lagging strand problem is usually not as deleterious as a leading strand problem by virtue of the repriming mechanism. And so I wanted to test this using a methodology that I had in the lab known as DNA molecular combing, which is a way in which you can visualize through the single molecule DNA synthesis and replication dynamics. How this works is it leverages different cross-reactivities of primary antibodies to different nucleoside analogs. So nucleoside analogs, of course, get incorporated anytime, in this case, anytime there's a T or a thymidine. And so you can pulse cells with them over short periods of time, wash them out, follow up with another pulse. And you can do the second pulse in the presence and absence of an inhibitor or a genetic condition. And then you can test at the single molecule what happens when you do that perturbation. After you've pulsed these cells, what you do is you embed the cells in an agarose plug. You perform a DNA extraction. This allows you to prevent shearing of the DNA. And then you stretch these DNA fibers out using a machine with a constant stretching factor onto a glass cover slip that's been silanized, so it's hydrophobic for binding the DNA. And you can stain it immunofluorescently, and you get something like this. And these are each, representative replication forks. And then what you can do is you can measure these replication, the DNA synthesis, and you can plot it out single cell or single lengths on a graph. And it's really important to note here that a shorter fiber indicates decreased leading and lagging strand synthesis. So any decrease in replication progression indicates that leading and lagging strand synthesis are both decreasing. Shorter fiber can also mean double strand breaks. So I did this simple assay and inhibited cells with pol alpha inhibitor. And as you can see very rapidly, with just 5 minutes of treatment, you see a decrease in replication fork speed. These are each individual dot is one of these replication fibers. And as you can see, this continues on 30 minutes and 60 minutes, indicating that when you inhibit pol alpha, it slows replication. So this led to the question, why? What is exactly happening? Because, yeah, I'm inhibiting pol alpha, but some replication is still going on. That first question is, how is that happening? Specifically with this substantial amount of single-stranded DNA that's occurring. And I thought, well, maybe one of the things is this isn't actually slowing. Maybe this has to do with fork collapse. So I alluded before that shorter fibers can indicate a double-strand break. And so I decided to test this. And by common assay, which is the gold standard for indicating double-strand breaks, overexpressing RPA, which has been shown to be exhausted in pol alpha inhibited conditions, and by knocking down structure-specific endonucleases, I can tell you that this slowing has nothing to do with double-strand breaks. It's something intrinsic to the replication fork. So that was ruled out. So next, I thought it would have something to do with fork reversal. Why? Well, as I said before, fork reversal is this process in which it's a dynamic process in which the fork can reverse and be restored. But also it's been shown that fork reversal can be induced by genotoxic stress, such as hydroxyurea. So hydroxyurea depletes free nucleotide pools, it slows replication, and it induces fork reversal. Also, it's been shown that fork reversal can slow DNA synthesis, and it can be relieved by knocking down several of the fork reversal proteins that have been characterized. I also wanted to finally rule out that this was not double-strand breaks, and what we do know is that a stalled or persistently stalled fork that's reversed can lead to double-strand break formation. And so I knocked down RAD51. I knocked down, which is a regulator of fork reversal. It's not a promoter protein. I knocked down ZRANV3, a motor protein, HLTF, and SMARCAL1, which are the known suspects for fork reversal. And what I found was some of the slowing was due to fork reversal, but a very small percentage. So this was not really satisfying to me. It was the fork is slowing, why is it slowing? And yeah, it could be because it's this part of the replosome, core component of the replosome. But I thought maybe they had an opportunity here. And I thought, well, maybe it's just because there's actually leading and lagging strand coupling going on. When you slow one strand, it slows the other. There had been a few factors that had been discovered. Maybe I was on the pathway towards discovering this. And I'd like to very much tell you today that I was on that pathway. And I did not find this human replication fork factor that couples leading and lagging strand synthesis. But in fact, I found something else interesting about the regulation of the DNA damage tolerance pathway. And that was because I did this experiment. So I inhibited with pol alpha, and then I looked at signaling, and the signaling was odd. So here you have hydroxyurea treated cells or pol alpha inhibited cells. And as you can see with hydroxyurea, you see this rapid increase of phosphorylation. This is only for 30 minutes. Of CHEK1 phosphorylation with this downstream effector of ATR. However, if you look at RPA 33, S33, which is a direct target of ATR, you don't see any phosphorylation at all. And it's known that HU over short periods of time doesn't induce a substantial amount of single-stranded DNA. However, I told you before that CD437 treatment increases single-stranded DNA substantially, so you can see that here in RPA phospho S33. However, really surprisingly, CHEK1 activation was kind of low. It was kind of puny. And this was really interesting to me because the general consensus is if you have more single-stranded DNA, you have more ATR activation. And so I thought, well, what could be happening? And I thought about, well, maybe something that activates CHEK1 is blunted. And that led me to think about this protein called claspin. And I'll talk about claspin in a second. But what I did was I overexpressed claspin, which is required to activate check one. And now you could drive this phosphorylation. So here again, same type of blot. And now I have this increase in check one. It isn't necessarily to the levels of HU. I don't have it on the same blot, so I can't say it with that, but just, observationally, it's not. However, I could drive this check one phosphorylation. So what is claspin? Well, we know it's required to activate check one. It's required for ATR dependent checkpoint activation. And then, of course, check one has a series of downstream targets. But claspin is also important for maintaining DNA synthesis. And so there was a study done in Keith Keldicott's lab that showed when you inhibited claspin, you get shorter replication fork fibers. And so in this case, I thought, well, let me overexpress claspin. I can recapitulate some of the check one signaling and look and see what happens. And really interestingly, even though I was inhibiting a core component of the replosome, I could drive replication fork progression. So here again, you have the same assay. It's plus and minus CD437. These are normal cells or those infected with empty vector. And now when I overexpress claspin, you can drive replication fork speed. Of course, it's not a full rescue because we are completely inhibiting alpha. But if you now add a mutant that can't interact with check one, you no longer see this increase. Further, if you do the same experiment, instead of overexpressing claspin, but now you inhibit check one with a check one kinase inhibitor, you no longer see claspin's ability to drive replication fork speed. So this leads to this model in this bizarre system in which ATR is active. You drive claspin, you overexpress claspin, you can drive check one phosphorylation, and you can increase fork speed. So I was trying to think like, well, what could be happening? How could check one be driving fork speed, or claspin for that matter? And I wanted to know whether this regulation was specific to this bizarre system or inhibiting pol alpha. So I thought, well, maybe what it had to do is something involving prim-pol. And this sounds like it's out of nowhere, but the thing is that I had tested fork reversal already, and there are so many TLS polymerases. And so I thought, if I knock down or look at one thing, maybe this is what's happening here. So first, what is prim-pol? I said before, it's this DNA-dependent polymerase that has de novo DNA primase activity. It reprimes inducing single-stranded DNA gaps, and it efficiently bypasses lesions such as A basic sites in 8-oxo-G and G4. So sure enough, when I overexpress claspin in this system, and now I knock down prim-pol, it's important to note that the absence of prim-pol you can no longer drive replication fork speed, indicating that prim-pol is, in the absence of pol alpha, can drive the replication fork, but only when claspin is overexpressed, as if claspin is sort of acting as a breaker activator. Importantly, this faster synthesized DNA contains persistent single-stranded DNA gaps as well. So there's a modified version of the DNA molecular combing assay that allows you to visualize whether there are single-stranded DNA gaps. And if these forks are faster, they have single-stranded DNA gaps. So I wanted to know whether this was specific to CD437 and claspin overexpression, this really strange model, or perhaps it was important in other scenarios in which prim-pol is active. So how does check one regulate prim-pol? I scanned the structure of prim-pol, and what I found was, really interestingly, there was this unstructured, uncharacterized region of the structure that has and contains a check one phosphorylation motif. And through a kinase assay in vitro that can show that phosphorylation happens, and SciLac IP mass spec, and a phosphospecific peptide antibody that does show it happens in cells, I found out that effectively in all other contexts in which prim-pol is active, this is during cisplatin treatment, a cancer therapeutic, ultraviolet treatment, and hydroxyurea, that prim-pol is active and is phosphorylated at serine 255, and that this serine is required for prim-pol activity. So how does this work? Well, when check one is there, you have prim-pol. So what I did is I took knockout prim-pol cells, and I complemented them with either wild type prim-pol or mutants. So if check one is there and you have prim-pol there, you can get repriming. So you can have a faster fork speed in the conditions in which prim-pol is active. This includes ultraviolet treatment as well as hydroxyurea and cisplatin. Now, if you introduce a mutant that is phospho dead, you get no repriming, you get slow fork speed, and it's inactive. Really interestingly, if you now introduce phosphomimetic mutants such that Primpol is now constitutively active, you get faster fork speed, but this comes at the cost of cellular fitness. But I wanted to really know what the mechanism was. Yeah, I showed that there was a direct phosphorylation, but I sort of hypothesized that perhaps something about binding of CHEK1 to this loop might, allow for Primpol repriming. And so I deleted this entire loop, so it no longer had the CHEK1 domain. And now Primpol acted exactly like the phosphomimetics, indicating that perhaps this loop is part of the regulation of Primpol. So we get this faster fork speed at the cost of fitness. So with that, CHEK1 phosphorylates. This was a sort of circuitous route, but it directly phosphorylates Primpol at serine 255. And why is this important? Well, targeting the ATR pathway is really clinically relevant. So there are CHEK1 inhibitors right now in clinic. And so if we really want to use them in clinic, we really want to know how and where they work. And the reason is because knowing where these inhibitors work really may help dictate in which patients we use them. So perhaps Primpol regulation or lack thereof determines specific outcomes because you're sort of pushing mutagenesis. And a really interesting example of this is that BRCA deficient cells have been recently shown to have persistent single-stranded DNA gaps. And indeed, BRCA deficient cells are CHEK1 inhibitor hypersensitive, indicating that this might be a nice way to understand treatments. So now I'm going to switch gears. This one's not so data heavy, and then I'm going to talk about the work in my lab. So I'm switching to this abasic sites, and abasic sites are really interesting because they're a common and potent polymerase blocking lesion, and they're caused by a host of things, including industrial chemicals, potassium bromate in food, chlorinated furanone in water supplies, lead contamination. And persistent abasic sites are mutagenic. They can form in duplex DNA, where they're subsequently resolved by base excision repair machinery. And unrepaired duplex DNA basic sites can be converted to single-stranded DNA lesions by helicase action, and can also be generated by cytidine deamination. And so I developed this system to induce abasic sites in human cells, and I used it to query some questions about this protein my postdoc lab discovered called hempses. So I alluded to this before, but hempses is this really interesting protein, and that it forms a strong covalent DPC at abasic sites in single-stranded DNA, preventing TLS and endonuclease digestion. Hempses null cells are hypersensitive to MMS, that's methylmethane sulfonate, UV, and IR. And what they all have in common is they can induce a lot of different lesions, but they also all in common can induce abasic sites. And hempses then subsequently ubiquitinated and degraded, and this DPC can also be stabilized by inhibiting the proteasome. And so when this paper came out in Cell, one of the outstanding questions was, is hempses responding to abasic sites alone, and is this the lesion? And so I aimed to test this with this construct that I made that allowed me to very rapidly induce ApoBec3A, the cytidine deaminase, into the nucleus, and asked the question whether hempses deficient cells were sensitive to ApoBec induction. And sure enough, they are. Here's a colony forming assay. This is plus and minus hempses sRNA. Some hempses sRNA are sort of more potent than others. This does have a defect in some cells that causes an arrest. But as you can appreciate, with the induction of ApoBecs with this 4-hydroxytamoxifen, you see a marked reduction in cell viability, as shown by this colony forming assay. And this was repeated in multiple cell lines. Importantly, there are more double-strand breaks as well. So mechanistically, what's happening is ApoBecs, some of the time when they deaminate, it results in a double-strand break in normal cells. But now when you knock out hempses, the double-strand breaks are much, much higher. And so I wanted to see, again, using my handy-dandy DNA molecular Kelming assay, whether perhaps hempses was important for maintaining fork progression during this ApoBec3A induction. So again, here's this assay, and as you can see, with the addition of A through A, you can slow synthesis. And actually, this is really interesting to me in that I actually expected that when I co-localized A3A to the nucleus, that there wouldn't be any effect on replication, being that it's been shown that in a basic site that's preferential to the lagging strand gets easily reprimed by the repriming polymerase. However, that was the case. And a really cool experiment is that there's this UNG inhibitor that's a derivative of phage. So it's a small peptide that inhibits UNG. And when you now treat cells with a UNG inhibitor, you don't see any slowing. So this indicates that the slowing is because of the basic site and not the uracil. Now you do this with HEMCs, you see substantial defect in replication fork progression that can be wholly rescued or nearly wholly rescued with UNG inhibitor. So in that paper, in that Cell paper in 2018, one of the really interesting observations that they made was they used this unbiased proteomic screen called iPond, which was developed in the Cortez lab, and I'm going to be using heavily in my lab, which is this really interesting and elegant way to discover what replication fork proteins are recruited to the replication fork in different genetic contexts or during different genotoxic stresses. And so it relies on mass spectrometry and EDU click chemistry to be able to basically pull down replication fork proteins that are at nascent DNA, and then very quantitatively measure, and compare what replication fork proteins are there in these different genetic conditions. And an important observation that they made was when you knock down HEMCs, you saw a vast increase in TLS polymerases to the replication fork. Interestingly, in another experiment here, when you knock down Rev3, which is this TLS polymerase, in the HEMC's background, you see a great growth defect, indicating that perhaps these were operating in different pathways. If HEMC's is there, TLS isn't, and if HEMC's is not there, TLS is. So I wanted to test this using my molecular combing assay to see if perhaps this was sort of one of the possibilities of what was happening. So I showed you before that when HEMC's is present and you induce a basic sites, you get slower fork speed. Now, when you induce in the absence of HEMC's, you get even slower fork speed. And there were some reports that TLS polymerases, particularly pol-kappa, are kind of slow. So I thought maybe this was because of TLS activity at the replication fork, and we're actually driving mutagenesis or potential mutagenesis. And so now we do the same assay and you block TLS by either inhibiting pol zeta or inhibiting pol-kappa, you get a fast fork speed. But this comes at a huge cost, and that cost is that the cells die almost immediately. So basically what's happening here is the basic sites persist. They don't have a TLS polymerase to bypass them. And so double strand breaks form, and this results in a cell viability defect. So although I have more work ongoing in this in my lab, in my postdoc lab, the conclusions here: the HEMC's deficient cells are hypersensitive to nuclear A3A. HEMC's protects A basic sites from processing into double strand breaks, and the A3A induced A basic sites slow both leading and lagging strand replication. TLS polymerases zeta and kappa slow fork elongation in the absence of HEMC's. And the current work and other questions are: does HEMC's prevent mutagenesis? So I've developed a mutagenesis platform in human cells to test exactly this. And we do know that actually in human cells right now, it doesn't seem like HEMC's itself is actually preventing mutagenesis. But what it is doing is perhaps biasing that mutagenesis to one of the strands, which I think is really, really interesting. And I want to know how it does that, if it does. So what next? So I'm going to go back to this statement before. So we really want to know about this pathway choice, and why each pathway is utilized is really important because of the outcome. And the outcome can be important for the cell that we're looking, or the human, that is going through a disease like cancer. And again, what determines which pathway you use is not well defined, with the observation that not all replication stress is the same. So in human cells, it's relatively unknown what happens, if you have a lesion in the leading versus the lagging strand. In other words, context, again, is important. So my lab has three main projects. One is: how do leading and lagging strand stress responses and strand mutagenesis bias differ? And the rationale here is that due to the differences in leading and lagging strand replication, mechanisms operating the signal and repair replication obstacles on each strand should be different. And I have a substantial amount of data that suggests that this is the case. And the goals and hypothesis are to utilize multiple approaches to study the mutagenic and stress response consequences of a strand-specific stall. Testing the hypothesis that these replication stress responses are differential and differentially mutagenic. And then I have a third sort of minor project that's sort of following up on some of my PhD work, because I sort of think it fits hand in hand. And I know it seems a little bit wild, but it's how do viruses as genotoxins alter human replication? And this sort of came about because my PhD advisor sort of handed me a lot of his collection, and I thought this would be something really interesting to follow up. But I'm really sort of viewing this virus, HPV, as a genotoxin and looking at to see how it actually alters mutagenesis in this pathway that I've developed. And I can really use the same tools that I'm using here. I really want to test the hypothesis that HPV employs both high and low fidelity polymerases for its own replication. I'm not going to talk much about this today. So how am I going to do this? Well, I'm going to characterize the effects of selective lagging and leading strand replication stress using these tools that I've already developed, including A3A, inhibiting equal alpha. And then I've developed these, tagged degron systems in which endogenous pol delta and pol epsilon are tagged with an auxin-inducible degron. So you can add a small ligand, and it can be very rapidly, both delta and epsilon can be rapidly degraded. I have data that suggests that this happens within just 15 minutes. And then I can do things like IPON, which I'm going to talk about in a second, CRISPR screens, and then mutagenesis and strand bias screens using these genetic toolkits that I've been working on over the past year and a half or so. So back to this IPON, which is this identification of proteins on nascent DNA. It's this really nice quantitative proteomics approach to characterize the human replosome. So I've done several of these in the presence and absence of things like leading strand stress, lagging strand stress, ApoBec induction, pol alpha inhibition, and generated a substantial amount of data and targets to look at that may be important for specifically for or preferentially for leading and lagging strand stress. And so when you do this, you get, like I showed you before, you get this really nice curve that comes out and several factors. So what is an example of something like this that I found? So first, I want to show you my systems again. This is this DNA molecular calming assay. And if you inhibit epsilon, pol epsilon, you get a massive reduction in replication fork speed. If you inhibit alpha, you get a massive reduction in replication fork speed. And if you induce apobecs, you get the same thing. So this indicates that there might be some sort of tethering mechanisms, but also some polymerases that are filling in. I mean, we know that delta can fill in for epsilon, but in the IPON data set, when I knocked or inhibited epsilon, one of the highest recruited factor to the replication fork was the TLS polymerase Rev3L. This is not the case when you inhibit pol delta. So this indicates that there's something about pol zeta that can respond to leading strand stress that does not happen in the absence of delta. And so from the IPON, I got this target. Now I'm using this degron system. You can see again that if I inhibit pol epsilon, you see the replication fork slow. And now if I also inhibit with pol epsilon with this JREH06 compound, replication fork slows even further, and these cells completely die. They can't maintain, suggesting that when you have a leading strand obstacle, potentially pol zeta is a first responder. And then this feeds into this pipeline that I've developed over the last year, this mutagenesis pipeline. And I want to show you something that a part of this pipeline that's just really, really amazing. So I culture cells for about 3 months in the presence and absence of genotoxic agents or genetic conditions. And I can derive really, really clean genetic signatures. And I'm not showing you all of the data today, but this also includes things like topology, nucleosome positioning, and all sorts of other metrics that we can be using for signatures, for holistic signatures in defined cancer cell lines. So here are normal wild type cells that were cultured for 100 generations. And you can see that there are something on the order of 2,454 substitutions. And then I cultured these in the presence of the strong oxidizing agent, potassium bromate. And just as you would expect, you get a substantial amount of increase in substitutions, but also a derived signature that's distinct from what's seen in the cosmic database. So this is just to sort of show you that this pipeline is working, and we're soon to publish another paper about this and Hemces, indicating sort of the mutagenic potential of both Hemces and TLS polymerases. And then this little side project I have on the side, which is to characterize the human and HPV replosome during HPV infection. And this is sort of born out of the fact that there's a bunch of really small pieces of information that still are not known that I have the toolkits to test. And those are like what polymerases replicate HPV and how HPV actually drives mutagenesis beyond degrading P53 and RB. And I've recently performed an IPOND experiment in HPV positive cells as a proof of principle. And really interestingly, in this experiment, again, in samples that are actively replicating HPV, one of the most recruited factors to the human genome is polymerase eta. So we know already that HPV pulls host polymerases away from the host genome to its own for its own replication. And this indicates that perhaps in doing so, it depletes some of the normal polymerases from human cells, resulting in the use of TLS polymerases. So with that, these are my acknowledgements. I still have my past lab on here because I still have only had my lab for two months. A special thanks to all of my collaborators and the people at UW-Madison that have been just exceptionally welcoming to me. It's been really, really wonderful. I am soon, well, when it finally opened, I will be moving to the new vet med building, which is going to be really fantastic. And if anybody knows anybody or is interested in the lab, just let me know. I'm recruiting on all levels. So with that, I will take any questions. Hey, Dave.
Very general question. So I'm wondering whether any of the effects that you're seeing are sort of cell type specific. If you did the same experiment, you've made an A-basic site in two different cells, would you see dramatically different effects on how they are repaired?
Yeah.
Etc.
So this is something I was worried about. So I made that the answer is they're not dramatically different, but another part of the answer is that if they are more cancerous, so a prime example, if you do this in RP HTERTs, right, which are just minimally immortalized with HTERT, the effects of things like A3A are much worse. They're much, much worse. So just these induction of the basic sites, they're much more hypersensitive to that in general than something like HCT116, which are still diploid, but they are mismatch repair defective, So I've been cautious about that, right? Because I am sort of concerned about that because I feel like perhaps there's a prime example is U2S cells, right, which have huge amounts of copies. I'm worried that if I tested in them, I saw something really interesting that you wouldn't see the same thing at all in RPs because they're already strained for polymerase use, and they might already be engaging they're likely engaging TLS polymerases. So yeah, point well taken. And so the degrons right now are in a mismatch repair defective cell line, and my every intent is to move those into more as normal as I can get them into. I tried really, really hard to make mutants in RPs, these degron knock-ins, and it's tough. They don't have the driving force to make it through that editing step at the polymerase, right? So that's one of the things that's complicating, but I'm doing my best. Yeah.
For questions, one of which is when you have your education for rehearsal, you have like the fork now has two double strands of milk. How is that just started? Are they really double strands?
Yeah, so there's it's not in human cells, it's not well known. There's a bunch of replication fork restart factors. MRE11, of course, is important to some degree. There's some chewing back that gets the right polarity on the ends. But it's it's quite complicated. I feel like I should ask Jim in the front row. So the motor proteins are dynamic, and then you also can start to get chewing back of the nascent strands, and then restart can happen also by CMG helicase progressing. There's a new model in which, that's actually from the Cortez lab, in which CMG is sort of put into this bubble when reversal happens, so it is much easier to start moving forward in restart. But I mean, there's myriad factors that are thought to be important, and almost all of the data relies on these DNA replication fiber assays, which I think are extremely beneficial and important. But I don't necessarily know. They can't necessarily tease out the mechanism by which these things are happening. So yeah, so my answer is a non-answer, really. Yeah.
I was curious with things like alpha that it seems to inhibit the lagging strand or the leading strand. If you think that the dynamics you're seeing would be replicated when DNA is structured, so it's gone around histons.
Yeah, I don't know. I do know that the single-stranded DNA that's formed in the lagging strand does accumulate other types of structures, things like G4 quadruplexes, because of the virtue of the length, the substantial length, and those certainly impede the replication. So, because if I stabilize them, then the replication gets even slower. But as far as with histones go, I'm not sure. Yeah.
Which part of your research in your new lab are you most excited about?
Yeah, the thing that I'm most excited about is I'm doing this CRISPR screen with low levels of delta and epsilon inhibition to determine what factors are really important during the low levels of delta and epsilon inhibition, and then cross-listing that with this screen. The thing that I think is tremendously exciting to me is that there are polymerases also in my list, which I didn't show here because it's actually kind of hard to see, but that really are unexpected, particularly in the context in which replication is happening. So those are things that I'm really interested in following up.
Thank you.
Yeah, thanks.